that. Good afternoon. My name is Monaghan. My project was an investigation into the interactome of the tumor necrosis factor receptor superfamily members, P75 neurotrophin receptor, and death receptor 6. So, in case you're not familiar with the term interactome, that just refers to the set of proteins that interact with a receptor or protein of interest. So for us, I focused on P75 and DR6. Both of these receptors are functionally related, and they are both involved in the activation of a sublethal caspase activation in the nervous system. Through this pathway, they are responsible partially for some of the neuropathogenesis of Alzheimer's disease. So Dr. Barker, whose lab I worked in, is actually a world leader in P75 research. So Matt himself is here in the audience, so thank you for letting me work in your lab. So moving on from there, let's talk about TNFRs. So tumor necrosis factor receptors are a diverse class of single-pass transmembrane receptors that bind to TNF-alpha and TNF-alpha-like ligands using extracellular cysteine-rich ligand binding domains. Members of the superfamily have been implicated in a variety of functions, including immune cell function, neuronal cell death and survival, autoimmunity, and inflammation. So, one of these superfamily members is P75, which is actually unique from the rest of the superfamily members, as it's a receptor for neurotrophins, which are nervous system growth regulators, and they're also involved in synaptic plasticity. So, it's unique because they're receptors for neurotrophins instead of TNF ligands like most of the rest of the family. So, P75 and DR6 are both involved, as I said, with caspase activation in the nervous system. So one of the possible effects of that is amyloid beta-induced synaptic impairment, which is part of the neuropathogenesis process of Alzheimer's disease. So from a cell signaling perspective, we actually don't know a whole lot about how P75 and DR6 signal. So in my project, I use a proteomics approach in order to uncover the interactome of P75 and DR6, which is going to give us a better insight into how these receptors signal, which will potentially lead us to find some therapeutic targets for treatment of Alzheimer's disease and related neuropathogenesis. So, P75 is known to undergo cleavage by alpha and gamma secretases, which causes the release of its intracellular domain into the cytosol. Once it's in the cytosol, it's thought to have unique signaling properties from the full-length receptor. This has been well documented in the case of P75 ICD, but unpublished Barker lab data has also suggested that DR6 ICD undergoes a similar process. So in order to study these cleaved ICDs, I use modified ICDs with a myristylation modification, which is added permanently, and it's a 14-carbon fatty acid modification which targets the cleaved ICD to the intracellular phase of the plasma membrane. This actually mimics its biologically relevant context for signaling, as P75 ICD has been shown to be rapidly cleaved by proteasomes once they're active in the cytosol. So again, the overarching goal of my project was to produce high-quality interactomes of P75 and DR6 full-length receptors and the myristylated ICDs of P75, DR6, and TROY using a method called BioID, which will uncover the set of proteins that interact with these receptors of interest. It's important to note here that this technique actually doesn't confirm every interaction that shows up on the screen, but instead it gives a set of proteins with a statistically high probability of interacting with these receptors. So along with completion of BioID, I set out to validate the BioID method by confirming tetracycline-inducible expression of the BioID constructs, as well as confirming the correct localization of these constructs to or near the plasma membrane. So I did this using techniques called cell surface biostimulation and immunocytochemistry. So following this, I turned my attention to confirming a physical interaction between P75 and DR6 with a common interactor called REL1, which is a TNFR-like molecule. And this was actually picked up on a previous BioID screen of P75 and DR6 that was conducted by the Barker lab. So I will go through each of these steps in turn and explain my results. So for the bulk of my project, I used a novel proximity-dependent biostimulation method called BioID. BioID relies on the fusion of the construct of interest to a native biotin ligase called BARA. Now what BARA does is it combines biotin and ATP to form a cloud of reactive biotin. And essentially what happens there is when you add the biotin, any proteins that come in to interact with the construct of interest get permanently tagged by biotin. And therefore, we can isolate these interacting proteins using a streptavidin pull-down. A streptavidin binds with very high affinity to biotin, and therefore pulling down any of the proteins that were bound by biotin, so our interactors. And then we identify those interactors using mass spectroscopy. The crude data is then put through some statistical tests, as well as some databases of common contaminants. So we're only having the significant and real interactors in our final data set. So the way I initiated this was I generated a modified HEC293 cell line for the stable tetracycline-inducible expression of the BioID constructs. Once I did this, some of the plates were used in BioID, and some of the plates were used for the validation methods I did on the side. BioID itself, at this point, is actually quite simple. Following selection, the cells are incubated in either tetracycline or biotin, depending on their conditions. The BioID contaminant database, known as the CRAPome, is optimized for 24-hour construct expression. So I made sure that this was put in place by only exposing the cells to tetracycline for that 24-hour period. So following the incubation, I then washed the cells, spun them down, and I sent the finished cell pellets off to the lab of Dr. Brian Rott in Toronto for streptavidin pull-down and mass spectroscopy. I'm currently waiting for these results. So before I go any further, I want to give a brief sales pitch on BioID, as the novelty of my project actually comes from the use of this newly discovered and effective method of detecting protein-protein interactions. So first off, BioID can detect weak or transient interactions, which diverges from older methods which are only optimized to pick up very strong or permanent interactions. Second, BioID is known to detect more interactors, and it can pick up interactors that occur at a lower abundance in the cell. And third, BioID uses one of the strongest known biological interactions, that between streptavidin and biotin, which is about 100,000 times more strong than antibody-antigen binding, which is the cornerstone of older proteomics methods. So once I prepared the BioID sample, before sending it off, I want to make sure I had absolute confidence in this BioID sample. So I set out to do two means of validation. So my first means of validation was a principle called cell surface biotinylation, in which I confirmed the construct expression in the presence of tetracycline, as well as its localization to the plasma membrane. So to start this, I incubated in tetracycline for 24 hours to cause the constructs to be expressed. And then I applied NHS-LC biotin, which is a cell membrane impermeable biotin conjugate which can't cross the plasma membrane. That means only membrane surface proteins are labeled by biotin in this way. Following this, the cells are lysed, and some of that lysate is used in streptavidin pull-down, in which streptavidin binds biotin strongly and therefore pulls down any of the membrane surface proteins that also came down with biotin, which in my case include my BioID constructs. And then in Western blot analysis, I detect the BioID construct specifically using a flag tag, which is specific, or a anti-flag antibody, which is against the flag tag fused to the construct. So in the lysates of the full-length cell surface biotinylation, you can see in lanes 4 here and here that tetracycline has turned on the construct expression of BioID, which is what we expect. And then in the streptavidin pull down, we see for both of these, when we pull down for P75 and DR6 through the biotin pull down of streptavidin, we do see that the construct has been pulled down successfully, which tells us that those constructs were originally localized at the membrane. We also see in these lanes here that we do have some leak expression of the constructs in the absence of tetracycline. This is because the tetracycline inducible expression is through a type repressor system, and this interaction is not permanent, so the repressor can come off transiently and allow some baseline expression to occur. So this wasn't very surprising to us. And then these are the ICEs here. And in the lysates, again, we see in the TET plus conditions of all three of them, we see that we have induced the bio-AD construct expression using tetracycline for 24 hours. And then in the streptavidin pull down, in the fourth lane of each of these three, we see again that when we pull down for biotin, we also pull down the bio-AD construct. This suggests that these have been pulled down from the membrane as well. So interestingly, you may wonder, okay, the cell impermeable biotin is labeling intracellular domains. How does that work? The key to this is part of that myrstillation modification actually has a portion that sticks just outside of the cell. So that makes it accessible to the cell membrane impermeable biotin. So through these assays, I confirmed tetracycline inducible expression of the bio-AD constructs, as well as its localization to the plasma membrane. So as my second means of validation, I use a technique called immunocytochemistry, which gives visual confirmation of the bio-AD construct expression as well as its correct localization. So to do this, I incubated in either tetracycline minus or tetracycline plus conditions for 24 hours, and then the cells were fixed to microscope slides using 4% PFA and probed with antibody against the flag tag. The signal was then detected with a secondary antibody conjugated to a fluorophore called Alexa Fluor 488, which allows us to visualize the construct in green. I also used a hooked stain, which stains DNA and allows us to see the nucleus. So as you can see in these left panels here and here, when we are in the absence of tetracycline, we get little to no expression of the constructs. You'll note here that in ICD conditions, that we do have some leak expression as well, which is in line with the results that we saw in the cell surface biotinylation. But in the plus tetracycline conditions, we see that we suddenly have all this green, suggesting to us that tetracycline has successfully induced expression of these bio-AD constructs, especially concentrated to the periphery of the cell, which shows that they were correctly expressed and then that they made it successfully to or near the plasma membrane, which is just where we expect them to be. So satisfied with our validation efforts to make sure that bio-AD worked correctly, I then turned my sights to confirm one of the interactions seen on a previous bio-AD screen, that between P75 and DR6, with a common interactor called REL1, which is a TNFR-like protein. So I did this using a method called co-IP, co-immunoprecipitation, which pulls out intact protein complexes. So the principle of this is you express tissue proteins, you lyse the cells, you target the first protein. So for us, in both cases, it was REL1. You label that with an antibody, and then you pull down on that antibody, which using beads, which will pull down REL1 as well as anything in a complex bound to REL1. And then in Western blot analysis, we then use another antibody for the second protein in the complex we're trying to detect. If we get a signal here, this tells us that there's been a physical interaction resulting in a complex containing both of these proteins. So this is the P75 and REL1 co-immunoprecipitation. And you can see in the input lysates here that P75 and REL1 have only been expressed in the appropriate conditions. In the IPHA conditions, we pull down for REL1, and we also use an IgG negative control here. So in the IPHA, which is when we pull down REL1 on those beads, we see down here, this band right above the background band right there and there, that we successfully pull down for REL1. And then the significant band right here is lane 4, which shows us that when we pull down for REL1 and we use an antibody against P75, that we get a signal there. This shows us that we've pulled both of these proteins out of the membrane together in a complex. This is the DR6 REL1 co-IP, and we actually get very similar results to the P75 REL1 co-IP. As you can see again in the input lysates, when we use antibody against DR6 and REL1, we can see that they've only been expressed in their appropriate conditions. We also use a negative IgG control here, and when we pull down for REL1, we can see here that we've picked up REL1 specifically, showing that pull down of REL1 was successful. And in the key band I want to draw your attention to here, when we pull down for REL1 and we probe for DR6, we do get a clear DR6 band there, showing that, again, these proteins have been pulled out of the membrane together in a complex. So, in summary, what I've shown is first, the expression of the bio-AD constructs was successfully induced in a tetracycline-inducible manner. Second, the BioAD constructs have been localized to the plasma membrane, and in doing so, I showed that myristoylation modification of depleted ICDs is an effective means of targeting the ICD constructs to the plasma membrane. And third, following validation, I confirmed a physical interaction between P75D6 and a common interactor called REL1. Fourth, pending results of the BioAD screen will be used to produce high-quality interaction data sets for P75 and DR6. These will spur on the development of hypotheses for future testing, as well as the potential identification of new targets for targeted therapy against things like Alzheimer's disease. As far as future directions go, I want to draw your attention to the RAL1. This is a fairly new interaction that hasn't been studied too much. And although I've proven that these exist in a complex with P75 and DR6, we haven't functionally characterized this interaction yet. So that represents a potential area of future study. So finally, I want to acknowledge the members of the Barker Lab. Thank you to Dr. Philip Barker and Dr. Julian Gibbon for the oversight and support through this project. And thanks to the other members of the lab, some of which are here. Especially to Mark Cumming, who directed my project and put up with my incessant question asking for the period of this project, as well as to the lab of Dr. Brian Rott, who will be doing strep test and pull down and mass spectroscopy. And thank you to you guys for listening. Very appreciated. Thank you.
Let's have some questions.
I have a quick question. Is DR6 what I at one time thought I knew was called Diablo?
I'm not sure of a historical name, but I know it is death receptor 6. I think it's super family member.
It's not the same thing.
It's different.
It's a different protein, yeah. DR6 is pretty new on the scene.
Any other questions? Yeah.
So when you're talking about the biotinylation of an intracellular target, you talked about a piece of the myristoylation being on the outside of the plasma membrane.
Yeah.
I was just wondering what piece was sticking out.
Yeah. So essentially, it's basically a fatty acid modification, and some of that sticks out just a tiny bit on the extracellular side. That's kind of the level to which I know it at. I haven't looked into the exact mechanism, but that's how that intracellular protein is labeled. It's through just a tiny piece of the membrane targeting modification. Good question.
Why did you go with co-IP to validate a protein-protein interaction as opposed to
Yeah, well, BioAD itself is a proximity-dependent method. So other methods to validate a protein-protein interaction, some of the more commonly used ones are also proximity-dependent. So using BioAD, I've already shown that REL1 has it is close in proximity to P75 and DR6. I don't need to verify that with another proximity method. So the answer to that question is co-IP really gets at a physical interaction, which is what I've shown. I don't necessarily show that it's a dimer per se, but I show that there is a physical complex form involving P75 DR6 and this protein REL1. Yeah.
You mentioned that this class of proteins activates caspases.
Yeah.
And you're massively overexpressing these proteins.
Yes.
So do you think you have to be worried that as a function of doing the experiment where you're overexpressing them, that you're actually killing a lot of the cells that are actually containing the proteins?
No, that's an interesting question. I mean, we certainly saw our share of massive death events, but I suppose the real thing there is this is the working model we have right now, and I mean, we'd love to see this in neurons and things like that, but the BioAD system just isn't ready to be applied to those things yet. So I think the answer is future studies will need to look at that more closely and really kind of get to the bottom of that issue.
Well, the reason, just if I can ask a follow-on question, the reason I'm asking is the biotinylation of the intracellular domain that was a question a moment ago, that is a massive amount of biotinylation.
Yeah.
So I'm wondering if an alternative explanation could be that what is really happening is you're killing the cells and you have membrane floating around in which both the interior and external leaflet are being exposed. So you're actually directly labeling the solar domain of dead cell husks, basically.
Yeah, that's an interesting question, and I don't know if there's a way in the scope of my project to directly see if that's the case. Yeah, future investigations would need to really look into that and kind of just decide if that's a real factor there.
Was a great job.
Any questions?
Well, thank you very much.